nos introduzamos, I introduced you to the vocabulary and the early images of this complex thing called the cell. Now we get into biology. Intracellular aspects of the process of protein secretion. This was the title of the Nobel lecture that Palade gave in 1974. He got the Nobel Prize about 3 months after I came back to Brazil for this last 45 years. And this talk has been 45 years in preparation. I gave it shortly after he got the Nobel Prize, which was about 10 slides. And with time, I kept adding to it, and I gave it about every 5 years. And this is my way of working, is a mathematician would say, by successive approximation. This drives my wife and my collaborators crazy because I never finished. This morning I wanted to change the second slide. Well, in terms of the definition of cell biology, the question Palade asked is, what is the relationship between the structure of the cell, organelles, and their function in the secretory process? In 1954, this is 8 years after Palade arrived at Rockefeller, he actively recruited, and gave a postdoctoral fellowship to Philip Siekowitz, who was a collaborator of his for the next 40 years. Palade was a very straightforward guy. He had an interest in biochemistry, but Phil Siekowitz actually described the first cell-free protein synthesis system. And Rockefeller could, give fellowships, and he literally recruited him. And they began the first EM and biochemical study of the pancreatic secretory cycle, which is the next block I want to give you. This was an inclusive system. I use the word inclusive because he intended to account for everything in a cell experiment. One could do an experiment and pull out the nuclei or pull out the mitochondria. But differential centrifugation, inclusive, is much harder because things were always slightly cross-contaminated. So they got enriched fractions of organelles. And many thought this study, which began in 1954 and wasn't published until 1958, was the first modern experiment in cell biology, or the first experiment of modern cell biology. And they really integrated the study. This is the disruption of cells, and on the right we have the supernatant from every centrifugation. So This is a centrifugation. You got a fraction of the pellet, and you got the supernatant. And then the supernatant was centrifuged, and you got a new pellet, and you got a new supernatant. You can see that they start at low speed or low centrifugal field for shorter times, and they increased the velocity, and with time they got smaller and smaller things. So the first they got a nuclear fraction which had contaminating intact cells and fragmented cells and granules. The next thing that would come down. The zymogen granules, you'll remember, are the secretory granules, the product. This is a factory for making protein enzymes and exporting them. And the package is the zymogen granule. Then they got the mitochondrial fraction, and finally the microsomal fraction. And in the final supernatant, there were free ribosomes. This was done in the 50s with the equipment available. Now there are better and faster and centrifuges. This is the biochemical readout of these fractions: homogenate, nuclear fraction, zymogen fraction, mitochondrial fraction, microsomal supernatant. First they did protein distribution, and you see they were very rigorous. They added up all the protein of the fractions, and they accounted for the protein of the homogenate. So this is the power of being inclusive, but also the weakness of being inclusive, and that was cross-contamination. This is the distribution of RNA. About 85% is recovered. And this is the RNA protein ratio. And they're all 30 or less, 0.3 or less, except the microsomal fraction. This has a lot of RNA compared to the protein. And this is the fragments of the endoplasmic reticulum. This is a paper of 1958, which is 61 years ago. So this is all they were doing, the distribution of protein and RNA. They didn't usually bother with DNA because they weren't really interested in nuclei. Now here's the proteolytic activity. These are proteolytic enzymes, which are the majority of the export products. And they are basically as inactive enzymes. The assay is very general, not specific. So if you don't add trypsin to activate, trypsin activatable proteolytic activity, you get very little activity. If you add trypsin and pre-incubate, you get activity, and it is much more, showing that the proteases are inactive. This is the protease activity. So most of the activity, not most, about a third of the activity is in the granule fraction, 36%. And the other fractions all have some activity. But the highest specific activity is in the granule fraction. It's most activity for amount of protein. So this is more or less what we expected, but this is the data. Okay? Next. I showed you ribosomes that were attached and ribosomes that were free. This is a tracer, a radioisotope tracer experiment in which the animal received C14 leucine. First, the pancreas concentrated the radioactivity, and there was activity both in the attached particles and in the free particles. With time, the attached particle went down, but the free particles did not change. And this is not total activity. This is activity in a specific protein. So it wasn't just general protein synthesis. This is the synthesis in a protein isolated by chromatography. See how careful they were. So the pulse moved through the microsomes, the endoplasmic reticulum, but the free particles retained newly synthesized protein, or continued to synthesize. And this is an experiment done 10 years later, but it proves the point. It shows that the counts went first to the microsomes and then to the zymogen granule. So this shows that newly synthesized protein is in the endoplasmic reticulum, is in the microsomal fraction, and then the radioactivity is transferred to the zymogen granule. This is the beginning of the series of steps of what we call the cycle. This experiment is pancreatic slices, which permitted them to have a pulse. It's hard to have a pulse in a whole animal. Pulse. Okay? This shows the point that it starts on the rough endoplasmic reticulum. So, they've already shown some things by combining electron microscopy and differential centrifugation and biochemistry in an inclusive study. What they showed was that the granules had most of the secretory product, and that the flow was microsome or endoplasmic reticulum to zymogen granule. The simultaneous development of biological electron microscopy and cell fractionation by the same group, the same investigators, in the same laboratory, was a very important catalyst for the explosive growth of cell biology. This had been done in 2 different laboratories. It wouldn't have come so quickly and wouldn't have been so convincing. And early in the time, remember, we started in 46. The first meeting of the society was 60. By that time, new departments of cell biology were being started, not biochemistry and biophysics, cell biology already had its name, or they added the name cell biology to existing morphology departments, which happened in our medical school, or they created new departments. This is an aside. Actually, the slide is taken from another talk, which I'll make propaganda for at the end of this talk. As scientists, we always have to ask ourselves, how good are our data? Data are plural in English. How good are our data? Does it suggest? Does it imply? Does it show? Does it indicate? Does it demonstrate? And in fact, when we write our scientific paper, we have to do that evaluation because we have to characterize our contribution. And we have to do that when we read other papers critically. And this is important because this goes to the other point of when do you stop and write a paper? They had data that had never been obtained before, combining cell fractionation and biochemistry. It was all new, but it wasn't a complete story. It didn't have a beginning, middle, and ending. And this is because differential centrifugation has intrinsic limitation, the homogeneity of the fractions, and the fact that once you break open fractions, organelles, things get placed in different lugars. They absorb. The ribosomes are very acidic because they're ribonuclear protein, so they'll absorb basic proteins like ribonucleic. So the problems of differential centrifugation, the fact that they didn't have all the steps in the secretory process. Remember, they had microsomes to granules. And it seemed to me that rather than spend more time polishing these experiments, Vladi and his team were already, 5 years before, investigating the new methods for EM autoradiography. Cell fractionation pointed and fixed Algun's facts, demonstration. But a lot of it was imply and suggest. So the autoradiographic demonstration was actually started at the same time that the cell fractionation was. And autoradiography for the light microscope existed. It was technically very difficult, and you can imagine what it was like for the microscope, electron microscope image. This is a paper published in '64, but actually is earlier. Caro started this in about '58, but he got sick, had a neurological disease, and the poor guy had to stop. This shows some grains and a badly fixed tissue, and was an injection into the whole animal. if you look at where the grains are, remember, you have a section, you put on an emulsion, you put it in a gaveta for a month, and you hope it's good. You open it, develop it. If it's so good, you put another one in for 2 months. There was, on the basis of the counts, the suggestion that ergastoplasm is really the endoplasmic reticulum and surrounding cytoplasmic matrix. They couldn't get very high resolution. They had few counts. But it seemed to go from the endoplasmic reticulum to the Golgi into the zymatic granule. This is the secretory process out of focus. So this whole animal study obviously was eliminated because the goddamn animal diluted the isotope. How could you do a pancreatic in vivo study without the animal? Well, you took out the pancreas and you sliced it, and you made slices. And where you cut, the enzymes came out. But they were able to show, with Jim Jameson showed that they could get a true pulse. Importantly, the normal morphology was retained after hours of incubation. So they did the electron microscopy to show that the sliced system behaved well. With that, they could get much higher concentration and more counts in the pancreas. And now a picture of the partially zymatic granules, the nucleus, endoplasmic reticulum, and all of these funny things are radioactive decay. So now they have at the beginning of the pulse, the counts are in the endoplasmic reticulum. We think they're on the ribosomes, but we can't see ribosomes here at this magnification. This is a very impressive demonstration of the endoplasmic reticulum, and here is Golgi, and there are no counts there. After 7 minutes, the endoplasmic reticulum, where is it? Here is no count, and we have the counts in the Golgi, counts, the spots, the black marks. So the isotope is here. And this is the apical part of the cell, the top with all the granules. Now it's hard to see disintegration in the granules, but there are none. The next shows the disintegrations over condensing vacuoles, but it also shows apparently in Golgi, maybe a little left over, to confirm that it's in condensing vesicles. They disrupted the cell and isolated a granular fraction that had condensing vesicles in it. And all of these irregular shaped beans are condensing vesicles, and that's where the counts are. So it's endoplasmic reticulum, Golgi vesicles, which are now called condensing vesicles, and finally granules. And in the apical part of the cell, again, apical, top, 3 different cells connecting lumen and counts. So now they have the whole story, and the numbers are better. Number of grains counted, and we can see that it starts in the endoplasmic reticulum. This is 3 minute, the pulse time. 7 minutes, it's endoplasmic and Golgi. 17 minutes, it's endoplasmic and peripheral of the Golgi. 37 minutes, it's going to the condensing vessels, and finally zymatic granules, and finally lumen. Now it doesn't go to 0, it stays in the endoplasmic reticulum, which continues producing secretory protein. But you can see that the drop is very dramatic and consistent. And the acinar lumen had very little, mitochondria had very little, and nuclei. And this is sort of the background. So I call this a demonstration. When an animal on jejune is fed, the pancreatic granules disappear. And this was seen in the middle of the 1800s by Claude Bernard in the human pancreas and by the physiologists. This was physiology at that time. The granules disappear and some dense content appears in the collecting ducts. So the next question Palade and his group asked are, the granules really the same as the pancreatic juice? We know the pancreas sits on the intestine, and a tube, a duct, pushes the pancreatic enzymes into the intestine. Well, the pancreatic juice of a guinea pig is a little. So the next study was done with cow enzyme. First, I want to say a little more about me. Why did it take me 7 years to get a doctorate? It didn't. It only took me 4 years. But when I went to Rockefeller, I went to the laboratory of Moore-Stein, who invented amino acid analysis, protein separation, did the first primary structure of an enzyme. I was a big analytical biochemist. That was my interest. I was 20 years old. I spent 3 years there on a thesis designed to modify peptides to increase the amount of tryptic cleavage. This is pretty esoteric, but it was a good solution to a very important problem in sequencing. After 3 years, I had to make my own peptides because you couldn't isolate them in quantities, and you couldn't use DNA to synthesize them because it wasn't available. And I used liquid ammonia, and I alkylated, and was actually having a hard time. And I opened Nature one day after lunch, and the answer was there. It was a very elegant study that solved the problem, that converted cysteine into an analog of lysine and provided a site of cleavage. There was nothing I had done that could add to it. They didn't have jeito brasileiro at Rockefeller, so I had to start a new thesis after 3 years. And I didn't like Morenstein, who then won the Nobel Prize, and I was one of the students, and I don't really know what happened. I was well trained in protein chemistry. I was interested in Pilates, and I don't know who made this marriage, but somehow I got into Pilates as his student with my orientador from the protein chemistry lab. And this is my thesis. Okay? So I was a protein chemist in a cell biology to be lab, and I couldn't focus the microscope, which is not that easy. Now it's done automatically. So the question I asked, the question that we asked was, are the proteins of the granule the same as the proteins of the juice? Because we had juice. This is a picture of the not an inclusive, a fractionation done only to get good granules. And if you made them alkaline, you got membranes. And all the proteins were soluble. So it was this extract that I compared to the pancreatic juice of a cow, to cow-cow experiment. Now, bovine pancreatic secretion was one of the main areas of contemporary biochemistry. The isolation, sequence, catalysis, crystallography was one of the frontiers of biochemistry. I essentially linked that frontier of biochemistry to these poor little guinea pigs. And the experiment where they showed the new counts in chymotrypsin was using bovine methodology. Now I'll just compare the cationic proteins, these are the basic proteins of pancreatic juice with zymatic granules, and the broken line is ribonuclease. This is tubes. This is equilibrium ion exchange chromatography. Well, the ribonuclease, and same amount of protein, same amount of ribonuclease activity, and these little peaks are glycosylated forms of ribonuclease. So the cow and the granules I got by going to the abattoir at 4 o'clock in the morning to get pancreas, big kilos of pancreas, took them back to the laboratory and isolated the granule. Now, this ribonuclease represents 3% of the protein. So these little fellows are obviously 0.1% or less. So in chromatographic position, quantity, and even for these trace amounts, the granule extract behaved like the juice. These are two other enzymes, granule on top, juice on the bottom, chymotrypsinogen, trypsinogen, and this is an inhibitor, trypsin, that I added to prevent activation. Now all of these scales are the same. So when the two lines merge or overlap, this means the specific activity of the whole peak is the same, which indicates homogeneity. Not only that, the scale ratios were equivalent to the best chymotrypsinogen or trypsinogen described in the literature. So it means there was no other contaminating protein of this basic property. So we showed they were qualitatively identical. The same proteins were in both. And to the extent that biochemistry is quantitative, which is 5%, 10%, there was the same amount. So this confirmed and demonstrated the idea that the secretory products are in the zymogen granule, and from the zymogen granule we can go back and account for all of the UV absorbance, 95%, and about 80% of the protein. And you notice that there's 20% missing by activity or by absorbance, and they're roughly half acidic, half basic, and half acidic. So this suggests that in the granule, they are macromolecular complexes, which reduces the amount of water. The activity is reduced. Anyway, the granule probably contains protein, cationic proteins that are interacting with anionic proteins, and that's why it's easier to get the water out. So, this data describes mixtures of proteins. The next question that was my thesis, and it was published in JBC, which Pilati liked. In fact, he only published in the JBC, the biochemical thing. So the next question is, do all the secretory proteins follow the same interorganelle pathway in parallel? Or are there, in other words, are there trypsin granules, amylase granules, or does every granule contain the same protein? Well, at that time, we didn't have the antibody markers, we didn't have the biochemistry, and they used the available methodology, and they had to invent another methodology. The pancreatic slice spontaneously leaked secretory protein because we cut it, so we broke a lot of cells. So they had to invent a new model for to replace the slice model. And they used, fortunately, the anatomy of the pancreas. This is a guinea pig pancreas filled with incubation fluid and in the connective tissue. And these are lobules of pancreatic acerous cells. So you can sort of expand the pancreas, and these little lobules were harvested, and they retain the morphology, and each one is a collection of cells around an interlobular duct, which is almost invisible here. So the whole animal went to the slice system and went to the lobule system. And these lobules weighed milligrams. So now they have lobules incubating, and there are at least two different stimuli systems, and all these things are plus or minus 10% more or less. All of the enzymes are secreted in parallel. They use full proteolytic enzyme, amylase, lipase, and DNase. And this is control. And this is everything secreted in 2 hours. This is a baseline secretion, which had the same composition as the secretion in response to stimulation. Looks parallel. However, this could have been 1% or 50% or 80% of the total enzyme. We can't tell because all of these are normalized at 100%, which is the cumulative value. Okay? So these guys were clever. This is Sheely and Pilati. They looked in the little lobule at what was left in a cell. So this was called discharged, which only used the total activity discharged. The next picture is release. And this is percent of the total enzyme in the cell. And these show that in 2 hours, 50% of the total enzyme is released. And this also continues in parallel. So the first one could have been misleading if it only accounted for 1% of the protein, because obviously the activity measurements are sensitive. So this says 50% of the protein in the lobule is discharged, and the proteins are, so this shows that 50% of the protein in the tissue is discharged, and the control is 5 or 10% in 2 hours. So they behave in parallel. This is one level of demonstration. And release counts what is. Here, other enzymes are 70% of the enzyme. This is ribonuclease, lipase, and DNase. Now, this whole thing had to be done in the presence of bovine plasma albumin. But the quantities were so small that, if you didn't put in the albumin, this line went down to this. All of these proteins were sticking to the glass in very small amounts. Okay, so the granule and the juice are the same. The secretion from the lobules is in parallel. So do the secretory proteins follow the same pathway in terms of organelles? It could be that they were collected in the condensing vacuoles from ribosomes, which are different, or different parts of the endoplasmic reticulum. So Alan Tartikoff developed a new micro system for the qualitative and quantitative description of individual guinea pig protein. This was not known. All the work could be done on bovine because you could get grams of bovine enzyme, and nobody was interested in the guinea pig enzymes. This is another student. He did isoelectric focusing, and this shows the cationic fraction, which is isoelectric between 8 and 10 or 11, and these are anionic proteins. He took a gel and cut it into 100 slices, and each slice is a fraction number of a isoelectric focusing liquid, no, sorry, this isn't a gel. This is liquid isoelectric focusing. They didn't have isoelectric focusing gels at that time. So about half of the protein is cationic, and when he spreads the cationic proteins, he gets different enzymes, and the isoelectric focusing doesn't resolve them. It's too close. And these are, I think, ribonuclease and chymotrypsinogen, and this is trypsinogen. So it separates cationic and anionic, but doesn't solve the problem. But if you put it on a gel, polyacrylamide, without isoelectric focusing, and you compare the granule content with the secretion content, and you have added albumin, you find out these are 4 dilutions this is 20 micrograms, 15, 10, and 5 micrograms of mixture. And you see that for SDS, acrylamide gel electrophoresis, you can't really tell the difference. So they look the same on gel. This is not a demonstration because it wasn't done quantitatively. But these are the isoelectric fractions, and you see these 3 proteins, 4, are basic. This is neutral, and this is acidic. And in terms of enzyme activity, it's roughly 50-50. even though he couldn't separate all the proteins, he had a very good way to measure mass. And that was with a radioisotope tracer. So he labeled all the proteins using uniformly labeled amino acids, put them on a gel, separated the gel into 100 slices, and counted, completely labeling them with radioactive amino acids is a kind of modern balance. You can weigh the amount of protein simply on the basis of the counts. Now, you didn't separate one from another, but you could look at it in terms of regions. Region 1, 2, 3, 4, and 5. Okay? Now, this is a check of the reproducibility of the method. You don't usually see this in a published paper. These are 10 microliter samples. These two, A and B, are duplicates, and this is 5 times as much, 50 microliter sample, duplicate. Cut the gel into 5 regions, and then plotted the percent of the recovered activity in 4 horizontal dots. Not so horizontal dots. This scale is expanding. So the first 2 dots are the duplicates of 10 microliters, and this is the duplicate of 50 microliters. So region 3 had 32% and 36% of the recovered activity. So this biggest dispersion is probably less than plus or minus 5%, because this scale only goes up to 40. Okay? So independent of the duplicates, and this one is the first one, and no proteins are in the first one. There's radioactivity, and that's because some of these things have sulfate on them. Not important. So looking at 2, 3, 4, and 5, biochemically identical. And now we can measure the mass of the material, or the radioactivity, which is proportional to mass. And this shows the system is valid. Now he's looking at the same system broken up into different components, 7, and this is where the major activity is. And this is the distribution. Again, from here to here is 10% of the activity. Okay? So these are close. These are less close. But what they're showing is that in time, 15, 25, 55, and 115 minutes, the proteins continue in parallel. And this is the composition by 7 regions of the gel, and we get, again, about a 50-50 distribution. And now that we're looking at something else, which is the proteins or the gel regions that come from three different stimulants, carbamylcholine and cerulean, give essentially the same. KCl gives a different composition, or somewhat different. No, KCl is similar. And no stimulant, basal secretion, is not very different from the others. So now we're seeing stimulated versus basal secretion with similar composition. And we're seeing the response to three different pharmacological agents giving the same compositions, parallelism. In the gels, they recovered the same amount, roughly 50,000 counts, and the secretion, obviously, is about 16%. And here we discharged 50% of the proteins. KCl discharged less, and this no stimulant. So everything they tried to do could not separate the individual proteins. So from this, we now have a more detailed, stepwise story of the secretory system process. Synthesis in ribosomes, adhering to the endoplasmic reticulum, and release into the internal part. We're going to come to that later. So the proteins are inside the endoplasmic, connecting to the Golgi, vesicles, accumulation in condensed vacuoles, storage at the granule, and release into the acinar cell. That is the secretory cycle at this level. And all the steps have been demonstrated. Because Palade was a scholar, he also looked at fibroblasts, granulocytes, macrophages, which all start the same way, common synthesis, segregation, transport, and concentration. And then things get different. Macrophages don't concentrate. They transport and discharge. Granulocytes do that, or they discharge. Storage is fibroblasts, and the whole thing is pancreas. So this is secretory process variations on a common theme. So this description of the newly synthesized protein on the ribosomes attached to the endoplasmic reticulum, then go through the Golgi, and finally exocytic secretion without breaking open the cell, completes the first chapter of description. This is sort of looking down at the autoradiography. But like all good science, it raises many more specific and interesting questions. First, how does a newly synthesized protein know, or how is it transferred to the inside of the endoplasmic reticulum? Then when it gets through the endoplasmic reticulum, not only how does it get the energy to move, but how does it know where to move and how long to move? In other words, this conduit system has to be able to, you have proteins going through a conduit system. How does the system address the protein? How does the protein, what information makes the protein go all the way to the granule and not stop in a vacuum? So the whole idea of addressing during this movement is really the next level. And I'm not going to go into it, but I'm going to show you the outlines of it. It's basically the CEPI number of a new protein. What CEPI does it have? How does it know which city to go to? How does it know which biro to go to in the city? This is what the postal CEPI has. So think of a letter that you mail to your friend in Belo Horizonte in the biro of Santa Maria. How can it get there efficiently? Preferably, how can it get there mechanically using information services? Well, obviously, the post office has ways of reading zip codes, CEPIs. In English, it's zip. Portuguese, it's CEP. And then addressing it to different cities and different byros. Okay? That's why I use the word addressed. Also, during the transit, we have post-translational modifications. We put on sugars, we put on phosphate, we put on sulfate. The whole world of post-translational mechanisms didn't exist then. They knew it existed, but there were very little details known. And then there's the question of energy. How do you physically move? These are obviously energy-dependent processes. Part of them are energy and part are not. The transfer into the endoplasmic reticulum doesn't require energy. Okay? So this is the next step. And this area was developed also by Pilati and his students and his postdocs and collaborators, which I'll discuss it a little more later. This is a paper of 66. Oh, doesn't it? Yeah, 66. Here they looked at ribosomes. They labeled an animal, and the counts were the dark solid line. They had ribosomes and medium, and that was the basic experiment. And you could see the amount in the medium rose and plateaued, the amount in the ribosomes rose and plateaued. Then they took the ribosome, or microsomes at that time, and treated it with deoxycholate. So here you have microsomes, which are called ribosomes here, and here you've opened the membrane system. The particles are still oriented on same structure, but there are no more circles. So the electron microscopy showed that they had opened the microsomes and the contents were now soluble. And you got this, soluble. So you went from this and this to this, soluble in desoxycholate. So the radioactivity, the newly synthesized protein, was already transferred into the interior of the endoplasmic reticulum. It doesn't say how, but it happened. Now, you could do the same experiment and treat with pure, the amount that was released was about 70 or 80%. Part of the newly synthesized protein stayed and pelleted with the ribosome. It was stuck, opening the microsome, didn't release it. Now you could incorporate and treat with puromycin. When you treat it with puromycin, the amount that is soluble is here. This is the control, is here and here. So puromycin releases newly synthesized protein, but like deoxycholate, only a fraction of it. Most of it. In contrast, streptomycin inhibits incorporation but doesn't release the protein. Now, this was done by Redmond and David Sabatini. David was from Argentina and a graduate student of Pilates. Redmond worked in the local Hemocentre and was a collaborator. Red Cross was across the street from Rockefeller. They had a picture of the ribosome, which was a little circle on top of a big circle. And that was the image they had. Then, this is published in 71, and what they have here is the messenger RNA, the new protein, which goes through the large part of the ribosome, carboxyl terminal, amino terminal first. The growing part of the amino terminal. So it's unidirectional and vectorial. And here you have more protein inside. Here you have more, and then all of a sudden it's cut and the whole protein is inside. So this is a proposal in 1971. And part of the information were these binding factors, which somehow interacted with the protein and the messenger RNA. They both had information. And this is the Blobel, Shealy. Well, Blobel was a postdoc of Plotty, and 30 years later, he got the Nobel Prize for the CEP code. He solved the whole problem and got the prize 30 years later. Sequence determination of 24 amino terminal residues indicated extensive homology. And essentially, the information was in the amino terminal part of the protein, which meant the same information was in the amino part of the RNA. So the information for vectorial transfer was in the messenger RNA and in the primary sequence. Now we know the primary sequence causes interactions, has information, which causes interactions between proteins, protein-protein interaction. So sequence information of amino acids has information, and sequence information of messenger RNA has information in addition to the information it has for replication. So it's in the amino terminal, and the amino terminal sequence is cut. And that's the first CEPI. And it comes from the messenger, goes to the protein, and interacts with the ribosome. So we now have the protein inside. This is a picture from Pilates' Nobel talk, which summarizes all we knew at that time, and we knew quite a bit. First, the protein is made using messenger RNA and goes inside the endoplasmic reticulum. Now, you remember that proteins have disulfide bridges, which are two cysteines which form a bond. Now, if you have a protein with 8 cysteines, you could form 4 bridges. In fact, you could form 105 different molecules with 4 bridges, mixing all of these up. So the information for the bridges, the disulfide bridges, is in the amino acid sequence and how it reacts with water and how the proteins fold. And there are enzymes that catalyze the search, catalyze movements of different kinds of sets of bridges. Thermodynamics says the most stable form will pop out. So one of the things that's going on in the endoplasmic reticulum is the correct pairing of the disulfides, because the protein is synthesized as a linear protein. If the protein happens to have 2 chains, it's because it was cut after the disulfides were formed. So the whole disulfide process happens in the endoplasmic reticulum to to find the right ones. Another thing that happens in the Golgi is carbohydrate is put on. We glycosylate. All of the post-translational modifications happen in different parts of the Golgi. So that's a lot of chemistry, a lot of information. We put on phosphate, we put on sulfate, we cut it here, we cut it there. All this cooking is all done in the process until you get to the secretory thing. So, finale. The scope of cell biology has become much wider than the demonstration that biological activities can be assigned to specific cell structures. I'm old and retired 15 years ago. I couldn't tell you about contemporary cell biology. This is about where I stopped knowing it well. Indeed, the artificial and conceptual boundaries that separated cell biology, molecular biology, biochemistry, and genetics are disappearing. And in fact, a contemporary, and this impresses me, a contemporary paper has all of these things, or 2 out of 3. And I helped my students until about 5 years ago publish papers, and we were looking at cancer cells and systems, etc. And the referees kept saying, that's a nice observation, but what is the mechanism? And why don't you have a genetic proof that your proposed mechanism? So things are much more complicated, and I honestly have not kept up with them. But one thing is clear, that biological sciences, which were very compartmentalized when I was young, just like the university is today, doesn't exist anymore. The university exists. The compartmentalization of areas doesn't exist anymore. These are all, it's hard. When I was young, you knew a difference between a cytologist and a biochemist. The biochemist was more intelligent, of course. But the biochemist lacked the appreciation of beauty and biology. He was rather too chemical. So we've seen this fantastic unification. Now I want to talk about post-graduation. I think I described about 20 or 25 papers over almost 30 years, which were the key papers. I mean, I've left some out. This is not a rigorous scientific review. What I want to point out is that all these people existed. I knew them all. And 1, 2, 3, 4, 5 were Pilates graduate students. Some stayed on for a short time. Some started as postdocs. Sequist stayed on at Rockefeller after Pilates left. And what I have either are processes or systems that was their job. They each had an area. There was no putting 5 students on the same problem to move it along quickly. So the FAPEST grant report we'll move along. Each one of these students really developed a system, slices, lobules, or analytical system, and then applied it to the problem. And this shows the mixture of ideas and methodology, and Palade was behind it all. This was only one part of Palade's interest. He had some other postdocs and collaborators doing other projects. He was interested in other things related to structure and function. Phil Siegowitz came on as a postdoc and died retired at Rockefeller. He then, Palade, remember, left in '73, and Siegowitz died about 5 years ago. Caro only did autoradiography, never finished it because of health problems. I worked on the secretory protein zymogen of bovine because that's where the methods were and what was an area very hot in biochemistry. Colvin was a collaborator who worked in the Hemocentre, and he worked on protein synthesis and vectorial transport. Jim worked on slices, autoradiography, and intraorganelle transport. He left in '73 and went to Yale, retired there, went there with Palade. And they started the new department of cell biology. David came from Argentina. He worked with a Di Robertis, who was a famous, who started cell biology in Argentina. He worked on vectorial transport as a doctoral student after getting his MD. Tartikoff was like me, Bachelor of Arts student. He worked on the guinea pig secretory protein and worked on parallel processing and cell fractionation, guinea pig. Gunther Blobel came to the United States in about '60 from East Germany. He was already an MD. He got his PhD at Wisconsin, came to Palade's lab as a postdoc, was in vectoral transport, protein targeting, for which he won the Nobel Prize. He stayed at Rockefeller until he died. George Shealy was already an MD, and he was a student, got his PhD. He worked on lobules, the system, studied parallel processing, and protein targeting. So Palade gave his students areas, didn't make them compete, but they all collaborated by putting it together. This is the finale to finale. The legacy of the group, Claude, Porter, and Palade, was cell compartmentalization. We have the beautiful nucleus, nucleolus, Golgi, endoplasmic reticulum, particles, green Golgi, condensing vacuoles, and exocytic secretion. We have mitochondria and other things, and cell matrix, I guess, skeleton. This is the legacy of the group. Pallad's legacy was bigger because Porter went to Harvard to work on plants. Claude went to Belgium to rebuild their university, and Pallades stayed on for another 30 years, 28 years. And as I said, he's my hero. He demonstrated the many different functions of eukaryotic organelles. He established the role of the endomembrane systems, the ER and the Golgi, in biological processes. His multidisciplinary vision of biological research, in particular his focus on the logic of organelles, defined a crucial turning point in our thinking about biomedical science. Cell biology has assumed a central role in the unification of experimental biology. Finally, I have a comment of Alan Tartikoff in a obituary he wrote for Pallade, and it is his open-mindedness and tenacity, along with his rigor and sense of intellectual elegance, has been remarkable. This is from a student of, and who came after me, but he's one of the few of us that are still alive. Pallade was very special. He had a classical education. His interest was in ancient history. He read about Romans in Latin, made sure that the name Endoplasmic Reticulum had the right derivation from the Latin. He was always accessible, never in a rush, always calm, and had everything thought out in advance. He didn't come in and make a decision. Okay, guys, today's we're going to do this, etc. He was truly a remarkable person and quite kind and nice. And what he did with the students really vindicated the ideas of the trustees when they insisted you need graduate students in a research institute. As I said, I do things by successive approximations. I gave this talk about last month, and I have already added 6 or 8 slides. I added sections, the zip code stuff at the end is new. Sylvia, Clarice, Lyris, well, Sylvia and Clarice have been with me for a long time, and they put up with me. I must be very exacerbating because I never finish anything. Lyris and Gustavo were my graduate students who helped me put this together in the form I gave it for my concorso for titular. And they're going to be very surprised when I send them a copy of this and they see where it went. Well, I hope that I've given you scientific information, but also stimulated you to think about what is the function of post-graduation and what you're doing in this process and what your orientadors are doing in this process. Thank you.